mitosis first. So, what we've been talking about is reproduction. And we had a cell, let's say it had 4 chromosomes. We knew how to make it into 2 cells that were identical to it, an exact copy. And that process was mitosis. And in mitosis, we took a cell which had 4 chromosomes. When it was just doing what it was doing, don't worry, that was just from dust. It was in G1, doing its job. From G1, it would go to S, make a DNA copy of all the DNA. So at the end of S, that cell with 4 chromosomes actually had 8 chromosomes in it. G2, we did last minute growth, and then from G2, go to prophase, metaphase, anaphase, telophase. Let's see, PMA, I need one more. T. And this was called mitosis. Oops, hit a button there. Giving us 2 cells with 4 chromosomes. So actually, from here to here, the cell had 8 chromosomes, which would mean 16 chromatids. Here we call it, there was still 16 chromatids. And when the cell split, 4 of them went to 4 chromosomes ended up in one cell and 4 chromosomes ended up in the other cell. Giving us, again, an exact copy. We started out with 1 cell all by itself. It became 2 cells. And that's great for making copies of cells. That's the way we want to make copies of cells. In fact, we said that we all started out life as a human stem cell. And after that, we went through this. And human stem cells have 23 pairs of chromosomes. 1 pair from mom and 1 from dad, which starts to give us an idea of why this won't work to reproduce an organism. Because let's look at where that human stem cell came from. So a human stem cell cell that has the ability to become a human has 23 pairs of chromosomes. There's 2 copies of one, number 2, all the way out to 23. And, 23rd pair is a little different, but we'll get into that later. So how did we get this human stem cell? Well, you've all had health class, and you know that we get that human stem cell by 2 cells coming together, an egg cell and a sperm cell, a human egg cell and a human sperm cell. So if this egg cell was a typical human cell, it would have 23 chromosomes, 23 pairs. The sperm cell would have 23 pairs. So 23 times 2 is 46. We'll leave it at that for now. This one would also have 46. And if 46 and 46 came together, we would have 92 chromosomes in this human stem cell, not 23 pairs. So we need to have a cell that has one copy of each and one copy of each. And that means these cells, the egg cell and the sperm cell, are different. And they're different in their chromosome numbers. So let's take a look at chromosome numbers in species. In most cells of an organism, there are 2 copies each chromosome. We're going to call these cells diploid cells. So let's just take an imaginary cell and say this is a whatnot. That's going to be the name of this organism. It's a whatnot. And a whatnot has 2 copies, has 1 chromosome, but there's 2 copies of it. So there's chromosome number 1, chromosome number 1. This is 1M and 1D. This chromosome number 1 came from its mother. This chromosome number 1 came from its father. So a whatnot has 1 chromosome, and there's 2 copies of that chromosome, a 1M and a 1D. The other kind of cell in an organism is called a gamete. And a gamete is a sex cell. It is only used for reproduction. So this is typical whatnot cell. 1 chromosome, 1 from mom, 1 from dad. The gametes are the sex cells. So whatnot sperm or whatnot egg are going to have 1 copy of chromosome 1, and it will either be the M or the D, not both. So we can then put 2 gametes together and get a stem cell that has the correct number of chromosome copies. So before we get into that, let's look at the steps of meiosis that allow this to happen. And then there's one other thing I want to talk about that happens during meiosis. So in order to generate these gametes, we use meiosis. And rather than drawing this in a circle, I'm going to do it in a linear path. So we start out with a gamete set. Gamete stem cell. And this is a whatnot, so it has a nucleus, and in that nucleus there is one chromosome pair. It's a 1M, which came from this whatnot's mother, and a 1D, which came from this whatnot's father. That stem cell is going to spend most of its life in G1 just hanging around waiting to become a gamete. From there, we'll go to S, and you will make DNA copy of all the DNA, which means you will have 2 1Ms and 2 1Ds when it's all said and done. From there, you'll go to G2, and after G2, you will go into meiosis 1, and meiosis 1 starts with pro, meta, ana, and telophase. But in prophase, tetrads are going to form instead of chromosomes. And a tetrad is all 4 copies of number 1, so 2 copies of 1M and 2 copies of 1D are going to form up in the same structure, and there will be 2 centromeres holding them together. A little hard to draw it with only 1 chromosome, but in metaphase 1, the tetrads line up in the middle. In anaphase 1, the tetrads split. That means some combination of 1M and 1D and 1M and 1D, and they will look like chromosomes. I shouldn't have drawn them apart. Yeah, let's do some erasing. Get this right. So some combination of the 2 1Ms and some combination of the 2 1Ds are going to split, and they're going to move away from each other. In telophase, and that will overlap. We're going to put them together, cytokinesis. We get 2 new cells. And those 2 new cells have half tetrads in them. And those half tetrads look exactly like the chromosomes we saw in mitosis. So for meiosis 2, in meiosis 2, we'll just follow one of them, we skip interphase. We don't make any more DNA. We go straight to prophase, from prophase to metaphase, from metaphase to anaphase, from anaphase to telophase. So in prophase, we have a half tetrad. In metaphase, the half tetrads align in the middle. And at the end of metaphase, the centromeres do split. I'm going to write that over here because it's very important. In metaphase 2, centromeres split. So now in anaphase and telophase, you're dealing with a quarter tetrad. From telophase 2, we're following 2 cells here. So you're going to get, I got too many lines here. 4 gametes, each with 1/4 of a tetrad inside it. So we start with 1 gamete stem cell. We end up with 4 gametes, 1 copy of each chromosome. 4 gametes, 1 copy of each chromosome, and that way the gametes from 2 different individuals So that way the gametes from 2 different individuals can join to form a stem cell. And that's why we can do, as humans, we would be able to take an egg, which has 23 chromosomes, combine it with a sperm, that has 23 chromosomes, get a stem cell that has 23 pairs of chromosomes. And that stem cell can then use mitosis to grow and develop. All right, so there's a couple of things I want to review on the board here, just this has been a little confusing. I hope you ask questions if you have them. I'll continue to do these chalk talks, and hopefully it will make more sense as we go. But there's some important things you need to remember. Diploid. Organisms Organisms have 2 copies of each chromosome. Haploid. Gametes have 1 copy of each chromosome. That way, 2 gametes can come together to give you a diploid stem cell. Now, the last thing I want to go over is something I didn't actually touch on it in this chalk talk, but I did mention it in the keynote, and that is this process of crossing over. There is no H in crossing over. So I said when a tetrad forms, you have a tangled mess of these 4 chromatids. So there's 2 that are copies, in this case we'll call this chromosome 1, 2 copies of chromosome 1 from mom, and 2 copies of chromosome 1 from dad, for a total of 4, which is why they're called tetrads. So let's, and if we just had this, those 4 gametes that were formed would contain, 2 of them would have a 1M and 2 of them would have a 1D. But that's not what happens. In crossing over, those chromosomes that we're going to make are going to be chromosomes that have exchanged materials. So instead of having, after crossing over, there's still no H in crossing. After crossing over, you have 4 chromosomes, and each one has parts of M and parts of D. So you don't have a 1M anymore, you don't have a 1D anymore. You have 1 chromosome that has some parts from M and some parts from D. You have another one that has different parts from M and different parts from D. A third one that has still other parts of M and parts of D. And a 4th one that is unique because it has new combinations of M and D. And this is really what the power of sexual reproduction and meiotic division is all about. It's all about diversity. New gene combinations. Mitosis was all about keeping things the same. Diversity is about meiosis, sexual reproduction, is all about getting the same but different. We want the same species, but we want new members that are different from the old members, unique members. So we're all human, but we're all very different, and meiosis, sexual reproduction, allows that to happen. We have variability introduced in crossing over. So you're not just getting a chromosome from your mother. You got a chromosome that had parts of your mother and your father on it. And 2, by combining gametes randomly, one out of the billions of billions of eggs your mom had, one out of the billions of sperm your dad had, came together. They all underwent crossing over. We get absolutely new combinations, always the same species, but always different from one another, unless you happen to be an identical twin. All right, so I will continue to post these. There'll be some more assignments coming up next week. Keep in touch, stay healthy.